Good day to all watching the video. This video is on countercurrent exchanger mechanism. As we have seen, there is a countercurrent multiplier mechanism which is the domain of the loop of Henle, which is performed by the loop of Henle. The exchanger function is performed by Vasa recta. What are these Vasa recta? These are the capillaries in the juxtamedullary nephrons, and their main role is to preserve the solute concentration. The solute concentration is built up with spending great energy, but that solute concentration can be just washed away by the blood. That is prevented by the vasa recta, which are specialized capillaries, and thus the wisdom of the body takes over and maintains the solute concentration, facilitating the action of ADH and helps build a concentrated urine. So we begin at the very basic. The kidneys have two regions. These are the kidneys. They have two regions, an outer cortex and an inner medulla. Outer cortex and an inner medulla. The nephrons in the cortex are called cortical nephrons, and they make up 88% of the nephrons. They have peritubular capillaries. You know, the renal circulation is a portal circulation with two sets of capillaries, glomerular and peritubular capillaries. The medulla, on the other hand, has a different set of neurons. They are called juxtamedullary neurons, juxtamedullary nephrons. So I'm drawing a line to indicate the boundary between the cortex and the medulla. In the juxtamedullary nephron, the renal corpuscle lies close to the medulla. Means the glomerular capillaries and the Bowman's capsule lies close to the medulla, and they have long loops of Henle, which we are concerned about. Long loops of Henle, long loops of Henle, and a special set of capillaries called the peritubular capillaries, and a special set of capillaries called the peritubular capillaries. Long loops of Henle, and a special set of capillaries is the efferent arteriole, and the efferent arteriole then leads to these capillaries, which are peritubular capillaries. But in the juxtamedullary nephron, these peritubular capillaries are called vasa recta. Vasa recta. They have long loops of Henle, and instead of the peritubular capillaries in the cortex, they have vasa recta. These vasa recta function as countercurrent exchangers and help preserve the solute concentration built up by the multiplier system. So the countercurrent multiplier system of the loop of Henle has expended a lot of energy. You can see in the thick ascending limb, the sodium potassium ATPase pump operates, and a lot of energy is expended to build up this solute concentration. Solute concentration means sodium, potassium, and associated anions, chloride and bicarbonate, plus glucose and urea, and plasma proteins. These are the solutes. This solute concentration is built up, and now it should not go waste. So, if it passes through the peritubular capillaries and just goes waste away, then the entire energy of the body is wasted. So, the body has a beautiful mechanism called as countercurrent exchanger. This is where Ernest Henry Starling's wisdom of the body is demonstrated at its best. The solutes enter the descending Vasa recta. The one which comes down is called descending, one which goes up is known as ascending Vasa recta. The descending Vasa recta, the ascending Vasa recta, solutes enter the descending Vasa recta. Now there is a big chance that the solute concentration could be lost, but they leave through the ascending Vasa recta. They leave through the ascending Vasa recta. They enter the descending Vasa recta, but they leave through the ascending Vasa recta. So the solute concentration is preserved in the medullary interstitium. Solute concentration is preserved in the medullary interstitium. So solutes remain in the medulla. Solute concentration is preserved in the medullary interstitium. Water follows an opposite path. Water leaves the descending Vasa recta and enters through the ascending Vasa recta. So water is lost. Now what is the total total osmolarity? Osmolarity is total solute concentration upon total body water. So the water decreases, but the solute concentration increases, like sodium, potassium, chloride, bicarbonate, glucose, urea, and plasma proteins. And this preserves the medullary osmolarity, and this preserves all the hard work done by the countercurrent multiplier system. Water decreasing also contributes, it increases the solute concentration in the medullary interstitium. So the Vasa recta function as countercurrent exchangers. Vasa recta function as countercurrent exchangers. Now there is, the kidney has two parts, a cortex and a medulla. The cortical nephrons contain the peritubular capillaries. The medullary nephrons, or the juxtamedullary, because they lie close to the medulla, have long loops of Henle. Instead of peritubular capillaries, they have Vasa recta. The solute concentration is built up by the countercurrent exchange mechanism, and then solutes enter the descending Vasa recta, leave through the ascending Vasa recta, and that helps preserve the solute concentration in the medullary interstitium. Apart from that, water also leaves the medulla, so water decreases, solute concentration increases, and as a result, a wonderful thing occurs. The medullary interstitium solute concentration is preserved, and hence ADH can continue to reabsorb water in the presence of aquaporins. Aquaporin 2 on the apical side of the principal cell of the lead distal tubule and collecting duct, and aquaporin 3 and 4 on the basolateral side. So the solute concentration built up by the countercurrent exchange system is preserved by the countercurrent exchange system, and that enables water reabsorption in the presence of ADH. It facilitates the action of ADH and thus a concentrated urine is formed up, concentration up to 1200 milliosmoles per liter. Thank you for watching my video. Have a good day and enjoy the day.